Welcome back to my channel. In this video, I will discuss about polymerase chain reaction, or in short, PCR. The basic principle of PCR, its steps, will also be discussed in a very simple way. I hope you would like this. And gradually, I will describe this technique in great details. But for this, you have to keep watching my videos. So let's start our today's discussion. PCR is a technique in which a short piece of DNA is copied several times from a very low amount of DNA. The original method was discovered by Carey Mullis in 1983, but that was slightly different than the current method we are using today. But the importance of his invention was soon recognized, and he received the Nobel Prize in Chemistry in 1993. This technique became popular, automated, and indispensable tools in biochemistry and molecular biology. After the discovery of thermostable DNA polymerase called Taq polymerase and a thermal cycler. Now, this technique is used in various fields like fundamental research, diagnosis, forensic science, biotechnology, environmental science, agricultural science, medical science, and lots more. The principle of this technique is similar to DNA replication. During the cell division, cell duplicated its genetic material, or simply DNA, and distributed it equally into each daughter cells. First, the two strands of DNA are separated so that the sequence of the bases can be accessed by the enzymes called DNA polymerase, which will make its copy. Inside the cell, DNA helicase and another enzyme separate the two strands of DNA using the energy of ATP. Then primers are synthesized by primase enzyme, and then DNA polymerase start new DNA synthesis using one strand as template in this way. But in PCR, the same chemical reaction is performed in the test tube, but in PCR, only a short portion of the DNA is replicated several times, not the whole chromosome. And for this, following chemicals are added in this concentration. The piece of DNA that will be copied multiple times is called the template DNA, because DNA polymerase use any of the two strands as template and read its sequence and add complementary nucleotides into the growing strand. For PCR, template DNA is generally isolated from the sample, and sample could be a human sample like blood, tissue, or it could be a microbes present in soil, water, or air, or it could be a fungus, virus, algae, higher animals, or anything in which DNA or RNA is present. And the region of the DNA that will be amplified repeatedly is called the target DNA. Two short oligonucleotides primers are also required that are synthesized in the laboratory. They are called forward and reverse primer. They actually decide where to start and where to stop. The primer which binds at the starting region of the target or the 5' end of the DNA is called forward primer. And other primer binds at the end region of the target or at the 5' end of the complementary strand is called reverse primer. Generally in PCR, 18 to 20 nucleotides long primers are used. Free dNTPs, that means all four nucleotides, A, T, G, and C, are also added into the tube cause these are the raw materials or building blocks for new DNA strand synthesis. DNA polymerase will add them into the daughter strand according to the information present in the template strand. A buffer is also required that will maintain a constant pH during the reaction so that the activity of the enzyme will not reduce gradually. The most commonly used buffer in PCR is this. We know that DNA is negatively charged and all four dNTPs are also negatively charged, so in order to minimize the repulsion, MgCl2 is added. A DNA-dependent DNA polymerase that is called the DNA polymerase is required because it will synthesize the new DNA strands. Initially, E. coli DNA polymerase was used as the replicating enzyme. Thus, after completion of each cycle, addition of new polymerase was required. E. coli polymerase is a heat-sensitive DNA polymerase. In 1988, the first use of thermostable DNA polymerase called Taq polymerase, which is isolated from the archaebacteria from hot spring, was reported, that leading the way to an automation of the protocol. The application of Taq polymerase to the PCR was the basis for the success of this technique. This reagent mixture is called the master mix. All reagents are mixed in a small tube, it is called PCR tubes. Then the tube is placed in a special instrument called thermal cycler. Discovery of thermal cycler also reduced the time of the process. Now we will see what is the function of the instrument. The machine will heat the tube at 95 degrees centigrade for 5 minutes. This step is called the denaturation. Then the tubes are cooled at 55 to 60 degrees centigrade for 30 seconds. This step is called annealing. And the last step, 72 degrees centigrade for 30 seconds to 1 minute, is called the extension. Then the machine repeated these 3 steps sequentially again and again. When all 3 steps are completed, it's called cycle. In typical PCR reaction, 35 to 45 cycles are there. Now we will see what happened in the tube during each step and how DNA is replicated. In this temperature, the DNA will denature, or we can say the 2 strands are separated. That's why this step is called denaturation. In this step, DNA start renaturation, or we can say complementary strands are again come close to each other to make the double-stranded DNA again. But the concentration of the primer are higher than the template DNA. So the primer will renature first before the 2 strands renatured. Here we can see that the primers are designed in such a way so that they will bind at the specific region of the target sequence. Suppose if you want to amplify this region only, so your primer should anneal in this and this region. In this temperature, the Taq polymerase start new DNA synthesis from where the primers are attached. All 4 dNTPs are attached one after another into the newly synthesized strands according to the information present in the template strand. You can see we have started the reaction with only 1 copy of DNA, but at the end of all 3 steps, 2 copies of DNA are synthesized. Now these newly synthesized strands can also serve as a template for the next cycle in this way. So at the end of 2 cycles, from 2 copies of DNA, 4 copies of DNA are produced. In this way, when 3 cycles are completed, 8 copies of DNA will produce. So we can say DNA is exponentially copied, and if you want to calculate the copy number after a particular cycle, you can use this equation, where small n is the cycle number, n0 is the initial DNA copy number, and nt is the copy number after n number of cycles. So at the end of 40 cycles, this much amount of DNA will be produced, and 40 cycles are completed within 2 to 3 hours roughly. Imagine how robust this technique is. Now I am going to solve one numerical problem that appeared in this examination. Here in this question, the PCR reaction was started with 3 DNA molecules, and you have to calculate the number of DNA after 5 cycles. So here small n is 5 and n0 is 3. We will use this equation to calculate this. And the number of DNA after 5 cycles is this. And here in this question, the PCR is started with 1 copy DNA. And after PCR is final, copy number is this. So we will again use this equation to calculate this. So in this case, 2 to the power n is this. Then we will take log in both sides and calculate the value of n. And here the value of n is 20. So 20 cycles are completed. I hope you will understand what is PCR, what is the principle, and how it copies DNA. In the next part, I will discuss in details how the temperature of those steps are selected and for how long and why and what will be the concentration of the each reagents in master mix. So that's all for now. If you like this video, press the like button, and of course you can criticize the content in the comment section. You can subscribe my channel if you like my contents and press the bell icon so that you will never miss any updates. Best of luck for your upcoming exam and thanks for watching.